Okay, so hello everyone. Welcome to today's panel discussion. We have actually more than 100 participants today, so I will make this short. So my name is Hong Miao, Associate Professor of Biostatistics and Data Science. I'm the moderator today. Today we're going to have Wei from UH to give us the overview. And then we have three speakers. Each of them have 10 minutes. We have to postpone all the questions until all the speakers done. So let's just leave it to Wei to start. Hello, Wei.
Yeah.
You can share your slides.
Yeah, I'm doing right now. And can you see my slides right now?
Yes, yes. Go ahead.
Okay, good afternoon. And welcome everyone to join our single cells workshop. And today's topic is mainly focused single cell technologies in immunology. And I'm an assistant professor at the Department of Biology and Biochemistry from the University of Houston. So here is my disclosure. And the unique things for immune-related study is that we are dealing with a very complex immune system, which consists with highly diverse immune cells. As shown in the slides, you can see that all the immune cells are derived from the stem cells, and from the myeloid progenitor cells will eventually differentiate into the immune cells, which contribute to the innate immune response, and the lymphoid progenitor cells will eventually differentiate and develop into the immune cells, contribute into the adaptive immune response. And because this is just overviews about the type of immune cells. However, each of these individual cells have the subsets. So that is giving highlights the need for us to use in the most advanced technologies to not only to check the abundance of these cells in the different tissue and organs, and also the function of these immune cells. So that is come to that the single cells technologies actually empowers immune-related studies. And at the early stage, when we want to do the single cells characterizations, mostly we use the limit dilutions method, which is that we diluting the cells to lower concentrations and then place these cells and 1 cells per wells to do the study. And later then we got the flow cytometries and the laser captures microscope. And recently we get the microfluidic systems and micromoleculator systems. All these technologies actually can help us to do the precise single cell isolation, and this advance significant empower our immune-related studies. So because of this advance in the single cell isolations, so right now, so we have a lot of different platforms to check the immune cells, the subsets and their functions. So for in order to determine the transcriptional profiling of the immune cells, we have the single cell RNA-seq, single nuclear RNA-seq, and specifically for immune systems is that we have for highly diverse TCR receptor and B cell receptors. So they also have the single cell TB cells receptor sequence. And for epigenetic profilings, right now we have the single cell attack-seq. And for proteomic profilings, we have the cytoph and flow cytometry to determine the different proteins expression level on the surface or inside the cells. And also is that recently, within the advance in the barcoding the antibodies, what they can do is that they also can do simultaneously evaluate or determine the both transcriptional and proteomic profiling So what they do is that they barcoding the different antibodies and then do the isolate single cells. And with these technologies, we can determine both the protein expression and mRNA expression at the same time. So currently, platform has cytic and abscisic. So all this is checking the molecular features of the immune cells. And we also want to check the molecular features of the immune cells with the spatial informations. And this is largely used the image platforms. So currently they has NanoStream provides the digital spatial profiles, and also we can use the imaging CyTOF to evaluate different proteins or RNA's expression on the tissues slides. Besides that, we also want to know the function of the immune cells. And traditionally, the typical one is we call ELISpot, which we can use the antibody and membrane to capture that the cells produce certain cytokines or soluble factors. And recently, we got the time-mapped image microscope and isoplex. So both of these ones can check not only the cells, the level, how many cytokines they can produce, also can check the cytotoxicity of the immune cells. So I just last slide, I list many different single cell technologies, and one take-home message is that all these technologies has their strengths and the weaknesses. So here I just use three most commonly used single cell technology as to compare the high dimensional flow cytometry, CyTOF, and single cell RNA-seq. So for the markers, currently flow cytometry only up to 25 protein markers, and this is limited by the fluorochrome diversities. As the advanced with we have more fluorochrome, so this number probably can go up. And the CyTOF right now is up to 35, and the single cell RNA-seq currently is around 1K to 2K transcript levels. And the cost is, as we all know, single cell is the most expensive, and the CyTOF is in the middle. For the sensitivities, the high dimensional flow cytometry actually can detect the protein expression at a broader range of levels. And the CyTOF is good, but might not as sensitive as the flow for certain markers. And for single cell RNA-seq, currently the platform, they are only very sensitive to abundant transcripts. And for the compensations for different channels, and flow cytometry is required and also very critical. CyTOF, they don't require the compensations, and the same as the single cell RNA-seq. For data processing, the high dimensional flow cytometry actually can handled by the research like us, and the CyTOF will probably need both immunologists and bioinformatics. And the single cells RNA-seq currently is mainly handled by the bioinformaticians, and hopefully in the future we have more user-friendly software to like the research like us can use without the bioinformatics assistance. And with that, I think I give Dr. Wang's platforms to introduce how he uses the single cell technology to answer the metabolic features of the immune cells in different disease settings.
Thank you, Wei.
Okay. Thank you, Wei.
Yeah. So Professor Wang, please share your slides.
Okay.
Yeah, so we did a very nice introduction for the technology. I found out it's a very powerful single cell technology. Probably almost about 2 years ago, we never touched it. We were focused more on the immunology study. We thought that we can handle a lot of stuff by the traditional method. It turned out that I could not be more wrong than that. So we found out that single cell technology is quite powerful in pushing our study of immune memory forward. And currently, we are studying. I have a lab at Houston Methodist to study immune memory against infections. So so everybody is affected by COVID-19 viruses. So and then hopefully most of us have taken the vaccines. So what happened to your immune system is that when you get the vaccine against the virus, either the proteins or the mRNAs, and your cells will produce antigens to activate immune cells. They will undergo dramatic expansion. And then the most of them, after do their job to good undergo expansion, hopefully fight the virus if you get infections, they will die off. And a few of them will survive with memory T cells and memory B cells and some other memory cells. So there are, why do we call them memory cells? Because they can live for a very long time. And they protect you for years to come, hopefully. And if they live for a year, then you have to get vaccination every year. If they live for 10 years, then you're protected for the next 10 years. So, so the formation and the life, the long-term persistence for memory cells are particularly important. And a few years ago, we found out that a process called autophagy is critical for the survival of immune memory cells. Particularly, we show that for the first time that memory B cells without autophagy, they will die off very quickly, so your vaccine will not work. So, so autophagy is essential for all the vaccines to work in order for your immune system to remember the infections and to fight it when you encounter it. And then we also accidentally discovered that the molecule called NIX is required for sequestration of mitochondrial alpha exon for degradation. And without NIX, the mitochondria stay outside. We use the first we use the red blood cell to study this phenomenon because it's pretty simple and the cells don't have a nucleus. And then this phenomenon also is true for nuclear cells like immune cells. And we recently finished a study for memory B cells, and we deleted the NIX and the BNIP3, two homologous molecules, and they function similarly. So we have to delete both together to see an effect. So when you delete them, then so this is a memory B cell, and if you look at the cells, you can see mitochondria outside in the cytosol. And if you delete these two molecules, the mitochondrial autophagy will not work, means your cells cannot get rid of the mitochondria by autophagy. And they were accumulated in the cytosol. And you can see a big rings of them and very elongated, they're confused together. And they're not good because the memory B cell were dramatically reduced. If you delete these molecules, means your vaccine will not work very well. And we did initially we did the biochemistry, and we were surprised to find out that the fat metabolic genes, a few of them here, are significantly increased in the mitochondrial autophagy deficient memory B cells. And then we figured that the more mitochondria are accumulated without autophagy because they cannot go into the autophagosome, and then we have more Krebs cycles to produce energy, and then the Krebs cycle intermediates, they will go to the cytosol to drive fatty acid synthesis, and we think that this is why these pathways are regulated. And we look at the different fatty acid synthesis genes, and they are all upregulated in the memory B cells with defective mitochondrial autophagy. And then we did the seahorse analysis, found that the memory, if you have autophagy deficient memory B cells, they have a much higher oxidative phosphorylation, and then they produce more fatty acids. And if you stain their the memory BCR, they have very small cytosol BCRs, TCRs, and then fill them with fatty acid. So these are, we joke, we call them obese memory BCRs, and they don't live very long. So why we are suddenly interested in single cell RNA, single cell technology, we found that when we after vaccination, we purified the B cells, and we found that there's one cluster of cells which are already becoming memory B cells in the first week. We call it it's in the class 1 here by single cell RNA-seq, and then they express these genes which are they are still not memory B cells. They don't have the phenotype, but the transcription profile is already converting them to memory B cells. So we call them memory B cell precursor or pre-memory B cells. And then we found out that they have already downregulated all the fatty acid synthesis genes at the early stage of memory B cell development. So when we target we inhibit the fatty acid synthesis genes, we found that it has a more dramatic effect on. It doesn't do anything for the memory B cells, but it's required for early activate B cells. So memory B cells need to be quiescent, live for a long time, don't make fatty acids, don't get obese. So this is how powerful the single cell technology telling us. And so we are doing our own single cell analysis, and we also working with people for help to analyze many public databases for the published memory cell information. The single cell technology, particularly the currently the RNA technology, and then protein technology, and also the spatial analysis will be vital for us to push our project forward to answer the important questions about the vaccination immune memory. We are focused on studying the mechanism for memory cell formation. So we are finding out factors which arise at the early stage of memory B cell development. So we think some of them are initiating factors. Then we need to find, in addition to autophagy, what are the mechanisms are critical for memory B cell survival and memory T cell survival. So we found that in order for them to survive, they need to downregulate their metabolic programs for fatty acid synthesis. And if you don't control that, the cell die very quickly. So the single cell technologies and the spatial analysis we just mentioned are very powerful technology which will allow us to make breakthroughs in this important direction. Yeah, thank you.
Okay, thank you, Jane. So now our next speaker is Nevin from UH. He's going to talk about single cell imaging. So Nevin, please share your slides.
Can you see my slides okay?
Yes, perfect.
Okay, awesome, thank you. My disclosures. Okay, so we already mentioned this at the start of the panel that we have many different technologies for single cell profiling. And I think we've already heard 2 examples of how single cell RNA-seq is being used, both from the experimental standpoint and the downstream bioinformatic analysis, for many different purposes, including predicting clinical responses, understanding how the disease evolves in preclinical models, and so on. So in parallel, CyTOF, flow cytometry was the original single cell technology. CyTOF came in, got rid of the need for compensation, cleared mass tags that allow you to separate the different antibodies, and you're able to design all of these panels to understand cell type identification, characterization of the phenotypes of the cells, and understand human immune cell relationships like Dr. Wang just showed. And eventually the goal is to correlate this with clinical features and then allow for patient stratification leading to responders and non-responders to therapy. But if you take a step back and look at the mechanism of action of biological molecules, biologics specifically, and antibodies, and more recently T cells as drugs, we can start at the bottom left center, and you'll see that, originally there were all of these antibodies that were thought to function just by blocking the receptor or the ligand on the surface, and then slowly moved on to the type 2 antibodies for anti-CD20 rituximab, where the antibody induced apoptosis independent of FC. And then you have all of the functional aspects of antibody-mediated responses. You have things like ADCC, which requires monocytes, and NK cells, and then you have ADCP, which requires macrophages. And so with the evolution of the checkpoint antibodies, where you're trying to inhibit these checkpoints, there's still some controversy on the exact mechanism and whether it involves the FC-dependent functions, specifically ADCC leading to depletion of Tregs, or whether it's clearly only a classical blocking of the ligand receptor interaction. But regardless, studying the functional embodiments of antibodies is a challenging task, right? And so just to emphasize the point, this is only a partial capture of all of the different functions of antibodies as we understand. And as I'm sure most of you recognize, that the FC neonatal receptor interaction based on the FC region of the antibody leads to its extended half-life as well. So almost all of the functions of antibodies are dependent on downstream effector functionality, whether it's FC gamma receptors, whether it's ADCP, and so on. Now, with the evolution of T cells as drugs, and specifically CAR T cells or tumor-infiltrating lymphocytes, CAR T cells, of course, have this happy marriage between an antibody and a T cell receptor leading to drugs. Nobody really understands the true mechanism of action of all of these. And so one of the questions my lab is very interested in, and has been for the last 10 years, is how can we study the functions of immune cells at scales across thousands of cells? And so if you look at the methods that are available for functional profiling, again, no need to go back into single-cell RNA-seq, excellent, but it doesn't have the concept of time. So the first three technologies that you see up here is single-cell RNA-seq, flow cytometry, and CyTOF. They have revolutionized the study of single cells, and single-cell RNA-seq is considered pretty standard these days. Most people can do single-cell RNA-seq, at least in a core facility, and they can understand, the cell type, expression of abundant transcripts, and so on. But these technologies are inherently limited in their understanding of time. And so, just like Dr. Wang mentioned, we've invented the concept of pseudotime, but pseudotime is good for a limited number of time points in which you want to understand differentiation trajectories, for example, but it's not suitable for functional profiling. We have developed a technology for single cell profiling based on microscopy that will directly allow us to assess the functional outcomes of the interaction between immune cells and the target cells. Vast majority of cases, it's tumor cells. And so because it's microscopy based, just about anything that will work with microscopy can be programmed into our assay. So we can look for subcellular organelles like mitochondria, lysosomes, and understand how trafficking of mitochondria and lysosomes can directly impact function. The nice thing is we can do this at scale, meaning that we can profile tens to hundreds of thousands of cells. Now, the concept itself is pretty simple, right? We want to be able to understand all of the different functions of immune cells, and specifically lymphocytes that they're capable of. So panel top left corner shows you phenotypic profiling. This is of NK cells, stratifying based on the expression of CD56 and CD16. You then want to be able to study cell-cell interaction. Does the immune cell interact with the tumor cell? And if so, what are the outcomes of the functional interaction? And then you want to be able to layer in cytokine secretion, which is an essential hallmark of almost all lymphocytes. And you want to understand the kinetics of immune cell secretion of cytokines. And lastly, it would be excellent if we can take advantages of the advances in single-cell RNA-seq and then come back and say, I want to be able to do single-cell RNA-seq on the same cell. So the key point that I hope to make in the next 5 minutes is we have built platforms on being able to understand all of these on the same cell without the need for doing correlative studies. So we can do functional profiling, phenotypic profiling, and single-cell RNA-seq on the same cell. And so why do we actually need, why can't we just do high throughput or high content screening, which is what everybody is used to? The problem is, tumor cells are adherent and they'll attach to the plastic. And so high content staining is fairly straightforward. You go back to the same location, the cell exists in the exact same spot, and you repeatedly profile it. Unfortunately, suspension cells and lymphocytes, for those of you who have looked at it under a microscope, these cells move, and they move a lot. They move about 10 microns a minute. And so studying the same cell as a function of time is a huge challenge just because of the movement or the motility of the cells themselves. And so the answer to that is actually very simple. How do you study things that move? Put them in cages, just like muffins in muffin trays. And our trays are exactly like muffin trays, right on cue at lunchtime. You throw these cells inside these muffin trays and they run and run and run. And there's one open dimension in which there's nutrient and media exchange. So we've designed a microfluidic chip that can encapsulate that has 20,000 wells that can encapsulate immune cells and tumor cells and image all 20,000 wells every 3 to 5 minutes. And we can go around this in cycles, right? We can image this for hours and days if necessary to understand the kinetics of the response. Now, the chip itself is spatially addressable, meaning even though we have 20,000 wells, any well within the 20,000 where we see functional interactions that are of interest to us, we have a robot that'll go retrieve that cell for us, and we can do downstream single-cell RNA-seq. And the last part is for those of you who do immune cell profiling, and specifically, T cell or NK cell mediated cytotoxicity. We have multiple effector and target ratios, which means that our chips are all internally controlled. You have target-only wells, you have multiple effector wells, multiple target wells, and so on. And this allows for robust statistics without the need for additional control chips. So basically, you load your two kinds of cells, you barcode them using fluorescent dyes, just lipophilic dyes, incubate them on a nanowell array, load the whole thing inside media. The open container, again, the open face of the chip allows for nutrient and media exchange. You add a marker for apoptosis so that you can track the dynamics of cell killing. Move this to the microscope with an attached incubator, and then off you go, image over and over again. So one of the problems we did not appreciate when we first started this was just the scale of the data itself. And so you have each well that's being shown here. You have many different kinds of cells. You have beads that understand or profile and quantify the cytokines being secreted by the cells, but you end up with 20,000 wells. That's 120 time points and 1 terabyte of data. No amount of manual annotation is going to help us. Thankfully, Badri Roysam, who's the chair of electrical engineering at UFH, their lab does image segmentation. And so they do all of the image segmentation and tracking. So the cells are automatically classified, and you're able to derive meaningful biological input and not have to sit and watch every single one of these videos. Now, with the advances in machine learning, we've taken advantage of the ability to distinguish the different kinds of cells and automatic calling of apoptosis. So we've modified our algorithms such that we can detect apoptosis automatically just based on the phase contrast channel. We're able to classify the different cell types as T cells, tumor cells, and so on. So I'll show you exactly one example in the interest of time on how we've used this. So we wanted to understand a very basic question in CAR T cell biology, chimeric antigen receptor T cells, specifically targeting B cells. So these cells in blue are CD19 CAR T cells, and these things in red are matched tumor cells that should be killed by the CAR T cells. And I'll show you two examples, one on the left, one on the right, by microscopy on what happens when these cells interact. So on the left, the CAR T cell was able to engage and kill all the tumor cells. You can discern this by the induction of apoptosis. The cells turn green. The apoptosis is also evidenced by the phase contrast channel, whereas this CAR T cell derived from the same donor, same population, conjugated to the two tumor cells, but basically did nothing. So one of the things I mentioned is we have a robot that can go pick out these cells to understand transcriptional profiling, and I can show you an example of what the robot looks like. So basically, the robot, I don't know why the video is glitchy. It basically picks out the cell from the well. You have image-based confirmation that the cell was picked up. And then it basically drops it in a PCR tube for us to be able to do single cell RNA-seq with it. So when we did differential transcriptional profiling of matched serial killer CAR T cells and non-killer CAR T cells, we identified transcripts that are differentially expressed between these two populations. We zoned in on TNFR SF9, which is CD137. And so we said, can this be used to improve the function of the CAR T cells itself? And so we transduced the CD137 ligand, which should stimulate the CD137 that gets upregulated on these killer T cells. Standard retroviral transduction. We manufacture the CAR T cells, and in a modified ovarian cancer mouse model in which we transduce the CD19 ligand onto SKOV3 cells, we show that the modified T cells that also co-express CD137 ligand lead to sustained and complete tumor control out to 100, 120 days. So this is an example of how we can use the combined information from functional profiling, identifying serial killer, non-killer CAR T cells, doing the link transcriptional profiling, and then using that information to manufacture the next generation of CAR T cells with increased potency. So basically what the high-level summary of the platform is that we want to be able to understand the dynamics of cellular function. Immune cells are based on their function. So we built a basic platform that can profile the function of immune cells across thousands of cells. At the same time, we're able to capture secreted protein using functionalized beads. And like I showed you with one example, we have a robot that will also pick out cells and do matched single cell RNA-seq or microfluidic PCR. And we integrate all of these information modalities at single cell resolution. So all of it is mapped onto the same single cell across thousands of cells. Acknowledgements, and I will pause and allow people to ask questions. Thank you.
Thank you, Nathan, for an interesting talk on imaging and muffins. So now it's the QA session, and we do want to start. And we do have a question in the chat channel. So
so that's a very nice question. The first question is asked for the cells that are connected or fixed at BSL-3 or BSL-4 labs. What are the recommended approach for the immune profiling? I recently did some COVID-19 related projects, study the immune profiling. Based on my experience, to follow the guideline of the BL3 and 4 lab, I haven't touched any project related with BL3 4. Mostly it's BSL3 labs. So for these samples, normally after the fix, they can move. You can transfer to BLS2's lab. So for these reasons, is the best approach is you can use the IHC, the immunochemistry stainings to study the fixed cells immune profilings like proteins expressions, and also the alternatively is that you can lysis your cells in the trisomes, and you can do the bulk, and after that you can transfer and do the bulk RNA or proteins analyze, the genomic analyze. And I think Linhua has the method which can do the deconvolutions to study the different immune populations. Dr. Jinhua, I know that you also do some COVID-19 related studies. Have you done any project related sample from the PSL 3 or 4? You're mute.
So we analyzed the blood samples in small quantity in pooled BRS2 plus facility. If we have to handle the virus, we have to use the level 3. We haven't done that.
So you haven't done single cells. You also say use
not single. Actually, I have a question for you. Can you do the spatial transcriptome studies, like after imaging the tissue on the slide and analyze the cells and convert to cDNA to RNA sequencing. That's what the text genomics have to work. Does your lab, can your lab do it? The
So
spatial RNA spatial transcriptome profiling.
Spatial transcription, that is mainly done by the nanostreams.
Yeah, okay.
RNAs. Currently, my lab has no, but I know a couple of labs have this, like MD Anderson's. Their platform has the nano streams based on the DSPs. That is called.
Okay.
Thank you. We have other questions piling up. So next one is the one from Lee for Nevin. Lee was asking, how do you know if the non-killer CAR-T is a non-killer CAR-T versus a killer CAR-T turned exhausted?
So the classification is based on the function at that time point, right? It doesn't account for the history of the cell itself. But that's it. There's a simpler way to answer the question of whether it's an exhausted CAR-T cell or not if we use the classical definitions of the inhibitory receptors, PD-1, TIM3, LAG3, and so on. And we don't see a difference between the killers and non-killer CAR-T cells in their expression of the inhibitory receptors at all. So it's unlikely that they're just functionally exhausted cells. So one of the things we have done, and I did not show in the interest of time, is that we've shown mechanistically it's not the exhaustion does not explain T cell failure to kill in vast majority of CAR T cell cases. The failure to kill arises from defects in granule polarization itself. So non-killer CAR T cells fail to sustain granule polarization. The lysosomes do not traffic towards the synapse.
Okay. Any other input from the panel?
So there's another question coming about the batch effect. So the question is, since the tissue are connected and processed through different protocol across lab or different single-cell RNA-seq method, how would you recommend different single-cell RNA-seq data or compare different data sets with different resolution? I think that is a good question for the bioinformaticians. So Hongyu, you have any insights about that? How to normalize the data across different batch?
Oh, actually, batch effect is always there. Okay, you have different, you have the different technicians or different equipment, or same technician, same equipment, but you do the experiments on different days. Okay, you see batch effects all the time. Okay, but statistically, we do have actually mature statistical model. And when I say mature, I don't mean that's perfect. Okay, we have some actually existing models and tools for that for batch effect removal. So I don't know whether we should consider that too much nowadays. But if it's like 5 years or 10 years ago, actually yes, it's a big problem.
So right now you are saying it's very reasonable that we can combine or integrate the single-cell RNA data
yes.
from the different experiments.
Yes, if your model actually take batch effects into consideration, then you don't need to worry too much about that.
So when we design the experiment, should we do we have any recommendations for us to reduce the potential batch effect, or you think that batch effect can absorbed by post occurrence analyze?
I believe Minhua may also have some inputs on that topic, but my simple suggestion, okay, or probably oversimplified suggestion will be try our best to have the same person to run the samples on the same machine. Okay, but definitely you cannot finish the jobs in one day, right? Sometimes you have a lot of jobs to run on multiple days. Then that's something we cannot control, but when we integrate data, we can take that into consideration. Okay. So thank you, Linhua. And we have another question from Ho Jia Kai. And the question is there any technique update for single cell sequencing by using fixed cells or tissues?
I think the most recent advance is a single nucleus RNA-seq. Probably you can use for the RNA profiling. This is personally I haven't used it. I don't know how robust this approach is. I don't know any of the panelists that have used this or has any comments about the single nucleus technology. Naveen, you have some comments?
No, I don't have experience with single nucleus, but it depends on what kind of fixation, right? Methanol fixation is compatible with almost every single cell RNA-seq workflow today.
Yeah, just this is come from the fixed cells, so we don't know how many RNA, what the RNAs result still remains in the samples. I don't have the hands-on experience on that so far. But I know I heard this technology has already entered the market. So.
Okay. We got another question for Neiman. So in general, what sample size do you aim for when analyzing the gene expression of a non-killer versus a killer CART? Is there much variation between single cells? Do you see single cell versus collective cell gene signatures? That's 3 questions. Okay.
Okay, so let me try and answer them one at a time. What sample size? We typically do a few hundred from each kind. So a few hundred, 200 to 300 killers, 200 to 300 non-killers, accumulated from at least 3 separate donors to also account for donor variation. Now, the second question is there much variation? There's absolutely a large variation, huge variation, especially donor-specific. Donor-specific populations are confounding factors in comparing killers versus non-killers. And so we do it both aggregated across different donors, and we do it in a donor-specific manner. And the last question is, do you see single cell versus collective cell gene signatures? I am not sure I fully comprehend that question. So if you are willing to please explain that to me, I can try and answer it.
I think that our participant audience can only type, right? So they cannot speak.
They cannot speak, only text. Okay, give me 1 second. Do you see single versus collective cell gene signatures? I apologize. I don't fully understand the question.
My guess is that if you have, we would just reply. So for example, if CAR cells are cultured together, does the gene?
Yeah, okay. Now that helps a lot. Thank you. So I think absolutely, right? We and others have shown in the past that T cells are social cells. They do better. Their differentiation, they're less differentiated when grown together, when there's T cell homotypic contact. Whereas if you grow them in isolated containers, they tend to be more differentiated. And specifically to overcome this, we only profile T cells that have been in the nanoval arrays for a few hours, typically it's 6 hours. And within that time frame, we don't see a major change in gene expression profiling due to isolation.
Another question, sorry, I have to jump in. So another question from Ling. How many biological replicas are needed?
Dr. Jinwang, you have any suggestion for your study? How many replicates you use for your single cells?
For single cells, so usually single cell, the core facility will sequence like a few thousand cells. And for real population, we try to get over 100 to get meaningful data we can calculate. So it's not a replicates, it's how many cells you sequence. So I think if you sequence, try to ask a core facility sequence 5,000 cells, you have over 100 cells in a particular population you are interested in, you can get very meaningful data.
And in my case is that, especially in the in vivo study, what I do is I pour the samples, at least 3 to 5 samples from different mice, and then harvest 10,000 from the pooled samples for sequence. Normally, we can get meaningful data.
Okay. That's actually a very useful and helpful answer. But that actually leads to another question. It's actually common, okay, when we use the single cell technology, okay, we do see rare population from time to time. I guess for that kind of questions, okay, the sample size calculation going to be a, more concern. So, but anyway, I just read it out for, for everybody to, jump in. So actually, I do actually have a question for Navin. So in your video, I saw that in the muffin cage, you have the cells moving around. Actually, I just out of curiosity, what factors drive them, moving around in that cage?
That is an excellent question for which I have very poor answer. So initially, we see T cell movement. I mean, T cell movement, if you go back to how T cells move in vivo, you need some kind of friction, right? You need friction mediated. There's no swimming that's been described for T cells. But our chips are actually functionalized with polyethylene glycol. So I really don't think that they're able to crawl on the surface, and there's no chemokines. It's just the random migration of T cells that is the movement you're seeing.
Yeah, but that gonna affect the chance they contact with each other, right? And
so again, but T cells move 10 microns a minute, right? And our wells are only 50 microns wide. Right, and so the ability to find target cells is not limited by their movement.
Okay, but I'm really wondering, will the pattern, their actually motion pattern will be different, be very different in real tissue and in your cage?
So we can do confinement. So it depends on what you want to study, right? And again, this is why I was careful in saying we're interested in understanding the properties of T cells from a therapeutic angle. So people who do 2-photon microscopy with CAR T cells, right? Say CAR T cells, throw them in mice, sack the animals, get the tissue and do 2-photon imaging. These people have shown that if all of the properties we study, right, T cell, tumor cell interaction, migration, killing, are all replicated in their model as well. So it seems like if these short-term studies of T cell biology in interacting with tumor cells is largely preserved. And so that's why we're careful not to study for days, which I said is, again, in response to an earlier question, it's confounded by the fact that T cell interactions are important for their survival as well. But short-term assays, it's very, very well conserved with how these cells behave after infusion, even in animal models.
Okay, thank you, Navin.
Of course.
I have a question for Levin. If we're collaborating with scientists at Baylor to develop a cure for HIV, and HIV is quite rare. It's in our treated patients, it's about 1 to 10 cells per minute, which are which have the HIV DNA integrated into the T cell macrophage genome. So, and then the NIH program officer want us to develop a method for single cell RNA-seq. I said, how do we study single cell in 1 minute? Why in a minute I have to sequence a million cells? So, is there a way to polymerize the cell to stain the DNA for the HIV and then sort it out and find out the RNA profile for the cells?
You should be able to do that. I'm blanking on the name of the author, but I don't know if you're familiar with INSEQ. They did permeabilization of cells
Yes.
to study transcription factors and then did single cell RNA-seq. But I'm not sure that 1 in 1 million is doable. That's at the limit of what even a flow cytometer can read. Right, and I'm happy to send you the in-seq paper.
Well, if 1 to 1,000, then we can study the
one in 1,000.
Not artery. 1 in 1,000 is possible, right? Okay.
Yeah, 1 in 1,000 should be.
So the system, if you can screen through like 20,000 cells in 5 minutes, then it's possible for you to go through some cells. Okay. So technically it's doable. Okay, maybe I email you to ask more details.
Yeah.
Other thing is so we also want to see where the reservoir is, for example, in the lymph node, in the tissue sections. So how do you get the spatial transcriptome study done for the sections?
Spatial transcriptome, the only good platform that I know if you don't want to do imaging cytometry is the Baylor core has the Codex platform set up. Is there anybody from Baylor here? I'm pretty sure Baylor core has the Codex platform set up. So you can do
Codex, what is
CODEX? Codex.
CODES. Codex.
Codex.
Actually, Dr. Richin from Baylor, and he can answer your question. I think.
There you go. So the Baylor core has the Codex platform set up, and you can reach out to them, and they should be able to do spatial profiling tissue.
Actually, I will probably ask the collaborator to do it because they're core. Thank you very much. Yeah.
That's for protein, right?
Yeah, that's directly for protein.
Yeah.
You don't need to do RNA. Codex is directly antibody-based labeling.
For protein, yeah. So can you do RNA with that? Probably.
No.
There's several, I think it's a good segue for the future panel. We'll talk about spatial single cell. So there's a lot of technology like a nanostrain, sort of not true single cell, but really like, 10 cells. But there's also true single cell ones like Murfish, and then also some single cell sequencing method. People are familiar with 10x Visium, right? Or future Visium HD, but also there's a DBIT and SlideSeq. So there's a lot of technology are ongoing. Some are getting commercialized, some are getting there. So we will have another panel specifically talk about this.
Are you, Dr. Chen, are you developing any technology in your core?
So currently the core is not doing anything in that front. The Visium, we tried Visium, and then actually I just reply email. Somebody may ask me about this. The resolution is quite low, right? So it's 55 micron. In some situations useful. So it's a cancer, will be useful. It's sequence based. But we are in the lab are doing we have Murfish, up and running in my current lab, and also we are working with DBIT. So those are close to be single cell. Murfish is true single cell, and then the DBIT will be like partial cell. So it's like it will be micron in resolution. So it's pretty good. So yeah, there's a lot of things going on, and I think Nick Navin's group are working with Resolve. Those are also single cell, panel. So yeah, there's some technology going on.
Okay.
Just follow up your previous question. So for the if you try to get one cell out of say 1 million, right? Very, very rare ones. It's almost akin to circulation tumor cells or circulation fetus cells, right? So if you have good marker, recognize that cell, there are microfluid based sorting method that are sensitive enough to capture 1 million or even less. So the key thing is the cell, you need to have some knowledge, you have some marker so you can see it. Right?
Yeah, the latent HIV infected cells have no marker. So
yeah, that's W. You don't know where to
hide in the cells. So the only thing you can find them is to look at their DNA, integrated HIV DNA. Or the other way you can do it is to activate them. You will find markers. Then all the transcription is changed, so you don't know what the original RNA profiling is. So it's but anyway, so the if you have some spatial technology developed, probably will be useful for the collaborator better to do the HIV study.
Yeah, I mean it's clearly where the field is moving to single cell.
Yeah, that's wonderful. Probably they will ask you for what kind of technique you can help them.
Yeah, so we're planning a panel sometime in the next. Stay tuned. Yeah.
Yeah, thank you.
Okay. So Niven, did you already answer our question, or you want to add something? That question for you, talking about the timing in the context of immunotherapy, and also the cell-cell interaction and different cell types, size, shapes.
Okay, the second question is easier to answer. The chip is agnostic to cell types, shape, and size. We can make the wells bigger, smaller, however we want. We've optimized them for immune cells, but we can do it for other cells. Has my lab studied any other cells? I'm sorry, if my training is in immunology, and so we've thought about only questions in immunotherapy. We've never done anything else. So we have not done, but there's nothing inherent about the platform that prevents us from adapting it to any cell type.
Okay. Any other inputs on this, or should I move on to the next one? So Lynn is asking whether it's possible to share some of the basic technology related slides. I think this panel discussion is recorded, right? And it will be, you know, freely available to participants. So, but I'm not sure whether, you know, all our speakers can share their slides, PowerPoint files with all their participants. But if you watch the video, you probably can get the information, okay, you need.
Yes, and that'll be on the Single Cell Omics website.
Okay, that's
if people want slides from me, I can send it to them. Some are unpublished, but it's all right.
Same thing with me. If you reach out to me and ask for specific slides, I'm happy to share anything.
My slides is open too.
Okay, thank all of you for being very generous. Okay, so I think we had a very good discussion and also presentation today. Actually, we still have 5 minutes left, but let me confirm. Okay, if you still have some questions, please ask right now.
Or you may never ask them.
Well, everybody got their 5 minutes back, so that's good. It's very good, you know, discussion. Thanks, you guys, for putting this together. It's wonderful.
Okay, thank you. And I guess that we can take that 5 minutes for lunch, right? Okay. Okay, then I think probably that's all for today. And you're actually welcome to ask further questions. You know our speakers' contact information, right? You know their name, their faces. So you can do that after the panel discussion. Thank you, everyone, for joining us today. And we have actually further panel discussions I don't know, in another 2 or 3 months, right?
Yeah, every quarter, yeah, every another 2 to 3 months, yes.
Okay, so please come back if you can. Okay, and thank you, everyone.
Thank you.
Thank you.
Thank you.
Thank you.
Bye-bye.